Okay, so this is our second session on Chapter 23, Organic and Biological Chemistry. So here we're taking a look at alcohols, another one of the functional groups, right? You may have heard this term, but it's here in organic chemistry, it has a very specific meaning. So this is a hydrocarbon that has a hydroxyl group, which is this OH function. It's like this part taken off water and attached to carbon. For example, ethanol, which is the drinking alcohol, is this small molecule. It's eth. It's got two carbons and an ol. The ol ending is the ending for alcohols. That's how an alcohol is designated, by adding an ol to the end. So simple alcohols can be thought of as derivatives of an alkane. So we start with ethane and we add an OH to the end, and now it becomes ethanol. So alcohols form hydrogen bonds and are typically, the lighter ones are typically soluble in water, and they have much higher boiling points than alkanes of similar size. Typically, adding hydroxyl groups is a way to get a lot of these organic compounds to become soluble in water, which is otherwise really difficult. As we were looking at things like alkanes, alkenes, these are like gases like propane. They are gases like natural gas methane. They are oily materials. They're not typically very soluble in water, but to solubilize even the heavier ones like hexane, which is around the weight of like gasoline, these are not going to dissolve in water. But by adding the functional group, the hydroxyl group, these can now be made to get hydrogen bonding and become soluble in water, have higher boiling points, etc. And you can name them with the location of the group. Again, this can get really complicated, but we're going to stick to simpler organic compounds for these examples. Note that you can have things like double bonds, rings, and hydroxyl groups all in the same molecule. So that's why our focus generally in this course is being able to identify these functional groups, being able to see, okay, this is an alcohol. And for the smaller ones, being able to see that this is a propanol. They've named this as a 1-propanol because there's a 2-propanol. There's a way to put that on propanol. You can also have other substituents like a 2-methyl-2-propanol. These are examples of things you should be able to name, and a cyclohexanol. So we take cyclohexane and we add an ol to it. So basically we're just taking the alkane, we've got an ol on here, and when we start our numbering like other functional groups, like the double bond and triple bond functional groups, we want to minimize the number of the functional group first before the substituents. The same basic rule still applies here. I talk about how methanol is also called wood alcohol, and it can cause blindness and death and really bad to drink. So ethers. Ethers can be thought of as, they say, as derivatives of water where you have carbon on both sides. You have an oxygen in the center and carbon on both sides. Another way to think of it is as an alcohol, but instead of the H, I put more carbon. And in fact, that's another way to think about how they're made. They're typically made from parent alcohols. They say they're relatively inert. They're often used as solvents. But they have an anesthetic effect, many of them. They have low boiling points because they don't have hydrogen bonding. And when you breathe them, they affect the viscosity of other properties of cerebral fluid. They tend to have this aesthetic effect is across the board in like all vertebrates. It's really weird. Even like fish get sedated by ethers. So it affects as a nervous system depressant, and they have this sedating effect. Diethyl ether was widely used before other anesthetics as a surgical agent to help people to dull their pain from like 19th century, Civil War era surgeries. But now that's been replaced by better alternatives. But bear in mind that when you use these as solvents in an organic lab, you need to use them with good ventilation, because while you're doing complex reactions, the last thing you want is to also get a little silly and sedated from the presence of ethers, which boil at low temperatures and may be present in the air. Doing these in a hood is important to keep ether fumes down. They're also highly flammable and can be explosive in certain cases, so got to be careful with that. Amines are, you can think of them as organic derivatives of ammonia. So if we take a parent ammonia and we add carbon groups to it, or we can think about it as the nitrogen analog of alcohols, is another way to think about these. The nitrogen analog of alcohols, right? So, except that we can replace it's like in this methylamine, this is like the nitrogen analog of an alcohol. But we can also put more carbon around it, and it's still an amine. This is still an amine in this instance. It doesn't get a special name like the ethers. these are still all amines. So as soon as I start putting carbon around ammonia, it doesn't matter how many are there. I don't need to have the hydrogen like the alcohol versus ether distinction. That doesn't exist for amines. All of these are considered amines. Amines have some nitrogen in them, and they have carbon on one or more sides of them. And many times they have hydrogen, but they don't have to. These are generally bases because of the lone pair on the nitrogen. And they're also generally soluble in water. These can also be increasing water solubility on conversion of an amine to its protonated salt, which is sometimes done as well. You take these amines and react with an acid to form a protonated salt. And this is important for making certain things soluble for delivery, certain kinds of drugs. So that's important and interesting. And now we'll talk about what are called the carbonyl groups. This is the whole group of functional groups that are characterized by having the the carbon-oxygen double bond present in them. We'll see them in aldehydes, carboxylic acids, ketones, and a variety and some other compounds as well. Carbonyl group compounds have similar properties because of this thing. So sometimes they'll just call this a function when it's not really one of the true functional groups. A carbonyl group or a carbonyl function is this C double bond O. And these are the functional groups associated with the carbonyl group, the simplest one being the aldehydes, then ketones, which are basically internal aldehydes. And this is kind of like the distinction between alcohols and ethers. Carboxylic acids go one step further and add another hydroxyl group to the carbon with the double bonded oxygen. Esters replace this hydrogen with a further carbon. And amides are an amine attached to the carbonyl. So all 5 of these are linked by the presence of the carbonyl carbon. Aldehydes and ketones are considered like the simplest versions of these carbonyl compounds. They have a double bonded oxygen attached to a carbon, either internally in the case of the ketone, where it's got carbon on both sides, or on a terminal end in the case of the aldehyde. We can think of an aldehyde as a terminal ketone, or as a ketone as an internal aldehyde. So examples of acetone is a ketone, properly named propanone. So that means we got 3 carbons and we got a ketone carbon in the middle, because you can't have a ketone on the ends, otherwise it's an aldehyde, right? Formaldehyde is methanol with 1 carbon, and that 1 carbon is the carbonyl carbon, and it's got hydrogen on both sides. Sometimes they also call it formalin or formaldehyde. Form is another term for 1, like meth and like mono, all of those mean 1. So keep that in mind. Where you see form, it means 1. Acete, again, we've seen that before, meaning 2. In this case, it means 2, 1 carbon on either side of the ketone. That's the thought process, not that there's 3 carbons in the compound, but that there's 2 carbons, 1 on each side of the carbonyl. That's how they think about those. So carboxylic acids, esters, and amines differ from aldehydes and ketones in that their carbonyl groups are bonded to strongly electronegative atoms. All 3 undergo carbonyl group substitution reactions, in which group we can represent as Y. So these can all have different kinds of substituent reactions where things can get replaced or added onto them. Carboxylic acids are really interesting. The simplest one that we typically encounter is acetic acid, also called ethanoic acid. There is also a formic acid. It's a potent irritant, though, and typically not used over acetic acid, which is the primary component of vinegar and very safe to handle and use. We have used it, if you've participated in any of our labs, you've used it many times as just acetic acid, and that's a carboxylic acid. Butanoic acid, benzoic acid, there are also complicated, longer other acids, right? These are named by replacing the final E of the alkane with oic acid for the systematic name. They also form a carboxylate anion when you remove the hydrogen from one end, because they act as an acid. They establish equilibrium. We've seen all this kind of stuff before. You can use acids plus alcohols to produce the esters, the derivative esters as well. That's a very interesting reaction you do in an organic class. Not a whole lot about esters. They're typically smelly compounds. They may also be bioactive in varying ways. Some of them are used to make synthetic fibers, but a lot of them are flavorants. Plants use these esters to produce various flavors, like the flavors of bananas, oranges, apples. Various things are the results of the presence of esters. And again, this is a very important reaction. You can also hydrate these esters back into their corresponding acids and alcohols as well. So these can be re-derived from a hydrolysis, a split with water. So the reaction that we saw here previously can also be undone as well in the presence of water. So amides, we have the carbonyl carbon bonded to a nitrogen. That's the key here. Example bioactive amides are things like acetaminophen in aspirin, the aspirin substitutes used in Tylenol and Excedrin. This is an amide. It's also an alcohol. It's also aromatic. These are other things you could notice just by looking at it. I wouldn't ask you to name it. You wouldn't be able to get the common name, and the other names would be very complex with multiple functional groups. But what I'm interested in is you being able to identify, like, oh, this is a hydroxyl group. This is an aromatic ring. This thing here is an amide. That kind of thing is what I'm really looking for students to be able to do coming out of this course, because it will accelerate your movement into an organic class. So we've got an amide here, and we can add water to this to cleave it and form an acid and an amine. Acids and amines can make amides through the reverse of this reaction as well. They say here to predict the products of this reaction. So this is an alcohol here with the carboxylic acid. And so we know those go together and lose the components of water, and the oxygen gets bonded to that carbon. And because we've seen that reaction. Similarly, this is an amine reaction. Amines, when they react with water, they replace the amine with water and lose the ammonia. At least in this instance, they're telling me some reaction happens. Not all of these are reactive, but amides like this are generally much more reactive than their parent amines, so keep that in mind. Here we also have a quick overview of biological chemistry. We're going to look at a few biomolecules and classify them. I'm not going to go into this in a whole lot of detail because it's kind of beyond the scope of this course, but we're going to see how biological systems utilize organic chemistry in order to function. The key here is that life does organic chemistry to survive, and we will see how processes in metabolism and other kinds of biological processes use organic chemistry and are driven by organic molecules with the same functional groups we've already looked at. They talk about digestion, and they talk about the breakdown of this. Again, I'm not really going to test this, but what I'm really looking at is the structure of things like acetyl-CoA. So we can identify that's a carbonyl carbon. We can make those kinds of connections, right, between the biochemistry or the biology that shows you all these molecules but doesn't really explain them to you, and now have a context of functional groups to help us understand why these molecules have the properties that they have, right? They talk about making ATP, which is how energy is exchanged typically within the cell, and how reactions are powered. And this phosphate, this is just phosphate, like the phosphate that we've seen before in other things that we've named. It's just two phosphates linked together. This is a complex molecule. This has some, alcohol functionalities here. This is some kind of ether here. This has some amine stuff going on in it. And we add another phosphate on here, and this is just some kind of helper thing with all these phosphates. But all these phosphates don't look too happy all bonded together. These little single oxygen bonds are probably not very stable, and that's by expectation, right? They're used to carry out and carry bulk energy, so the reaction and loss of phosphates is part of how they transfer energy. It's really interesting in a lot of cases are proteins, right? Proteins are made up of amino acid monomers. By their name, they have an amine function and a carboxylic acid function in the same molecule. So these are what we'll kind of call difunctional molecules. They're relatively simple, but they are difunctional, so they're a little more complicated than some of the stuff we've seen. But proteins made by living things are all made up of these little amino acid units, and they have different names that we've given them, and they go by those names. They don't go by any kind of systematic name. And amino acids can be brought together by the reaction between the carboxylic acid and the amine, an acid and a base. So really this is like an acid-base reaction that we've seen. And the formation of a new amide bond here, which here is called a peptide bond, where it links two amino acids together. You can repeat these on ad infinitum and create long proteins with all kinds of wonderful biological functionality. But the key point I'm pointing out to you is it's just an amine, and it's just a carboxylic acid, and it's an acid-base reaction where an acid and a base react to make this thing, which we can think of as like the salt, and this thing as the water, gets lost. And this forms an amide bond that is a peptide bond. That's what they call it. So essentially, we're looking at this from an organic chemistry perspective, whereas in biology, they'd look at it from like a biological perspective. But what I'm trying to point out to you is there's no magic. These are just the same functional groups we just looked at, just in a biological context. Also, all the biologically active proteinogenic, the ones that are used to make proteins, amino acids, are all alpha amino acids, where you have this one carbon, they call this the alpha carbon, to which both the acid and amine functions are joined, and then some side chain R, which tells you which amino acid it is. All amino acids have the same basic function, and this may be something I ask on a test or a bonus question, asking you to draw this to tell me, to show me that all amino acids have the same basic structure. You could have a beta amino acid, you could have another carbon here. It's just that living things don't use that one. That's not the one that's bioactive. But such things exist. Like any molecule with an amine group and an acid group is technically an amino acid from a chemical standpoint, but it's just not one of the 20 proteinogenic amino acids that we typically refer to as amino acids. And we can think about all of them as having some R group. Now, because they have four different things bonded to the central carbon, most amino acids are chiral, which means that they have a left-handed and a right-handed version. And only one of the handed versions is the one that biological systems use, because in order to make large chains, they need all the amino acids to be the same handedness so that they react best with each other. They like form a set. Maybe on a different planet they use the other set. They're not chemically any different from each other. They're just together, they react chirally in relation to each other. So most of them, aside from one where this side chain is just a hydrogen, which does exist, the rest of them are all chiral, and we'll see that when we look at the structures. So when we look here at our simplest amino acid here, hold up, let me get that one in here. Yeah, glycine, which I think of as like the simplest one. It's got a hydrogen for the R group. So this is like the simplest, of the 20 amino acids, because it's just a hydrogen. Alanine is just a methyl group. Valine is an isopropyl group. And you can see how each of these is a kind of, this is a sec-butyl group. It's another type of butyl group. So these are just chains like ones that we've seen before. Like this is some kind of ethyl group here, one carbon here, like a methyl group, plus the benzene ring. So you can see how these molecules have different functions. That's really what I'm hoping you focus on. I'm not looking for you to memorize all the names of all the amino acids. It's for a biology course. It's for a biochemistry course. But what I want you to get in this course is how the functional groups are, the basic ones are all the same, and the different substituents can bring different functional groups to the mix. Like they can bring in an amide function, they can bring in alcohol function, they can bring these kinds of sulfur-like alcohol thing functions, which we're not going to discuss in any further point, just to be aware that other types of functions exist. We're just talking about the basic ones. Lysine, you can bring an amine function to the list. So keep all these things in mind. Even though these are the 20 biogenic ones, you can make non-biogenic ones. They exist in some cases too. Non-proteinogenic ones. They have other purposes in other places, and you could use them maybe to make something that's not even associated with living things. So they show here, because amino acids can be assembled in any order, depending on which acid group forms the amide bond with which amine group, possible isomeric peptides increases rapidly as the number of amino acids increase. There are 6 ways in which 3 different amino acids can be joined, 40,000 ways in which 8 can be joined, and then they talk about here which the 8 present in the blood pressure regulating hormone can be joined. Many biological systems are regulated by the presence of proteins. Smaller ones they sometimes call peptides, and even huge enzymatic structures that are all made out of amino acids fundamentally. No matter how long the chain is, all non-cyclic proteins have an N terminus and a C terminus. So you'd have to put one in a circle if you wanted to get that out. Protein is written with the free amine end on the left, the N terminus, they call it by convention, and the free carboxylic acid terminus, the C terminus, on the right side. And they use 3-letter abbreviations for each of the names that are listed in the figures before. So they want us to draw the dipeptide al-ser. So I look up alanine and serine, and I just bond them together where they put the al first where the N terminus is and the C terminus. And this is just by convention, the way humans decided to, label the directions. But this is the basic idea. It's pretty simple in that sense. So what are carbohydrates? We can think of carbohydrates as like polyols, like having a lot of alcohol functions all throughout them. So they used to think of them as like hydrates of carbon, because when they burned them, they'd get the basic formula, C H2O as some formula, where the H2O would be some X, and this is like, and the carbon would be some X. So you get the same number of waters for every carbon, and that's where the name comes from it. They call these a large class of polyhydroxylated aldehydes and ketones called sugars. So when we look at the structure, we see, okay, this is an aldehyde. These are a bunch of alcohol functionalities. So we can see how these biological molecules are made of the same basic components as our simpler ones that we were able to look at and name and break down, right? Simple sugars are monosaccharides. Complex carbohydrates are polysaccharides. They talk about how they can react and form rings, and you can look at how you can put prefixes on them for, how many carbons are in the sugars and how many ones are in the ring and how many rings you get together. But again, like I say, my basic focus is on you being able to identify them as having functional groups and working like any other kinds of organic molecules. Similarly, this is some kind of ether linkage. This is some kind of ether linkage here between these two sugar rings. And the fact they form rings, these are just, like a cyclo version of a compound when they form rings with each other. And we see that the rings with each other they form is because an alcohol reacts with an aldehyde to dehydrate into an ester or ether functionality here. Lipids are organic molecules that are not very soluble. They have long chains of carbon. Generally, we can kind of think of them as a kind of biological gasoline. They're not typically made very soluble, and they have very long hydrocarbon portions. So they form animal fats and oils and cell membranes and all kinds of things like that, called triglycerides or fatty acids, varying derivatives. Some of them have acid functionalities on the end. Sometimes they're branched or unbranched. They have very long carbon chains. So in this sense, their reactions with oxygen, they take a lot of rotations and they can store a lot of energy. That's why I'm comparing them to a kind of biological gasoline to me. With long carbon chains like this, they're kind of like oil, like motor oils, or like diesel fuels. It's actually possible to do certain kinds of reactions with lipids to turn them into compatible fuels for diesel engines. That's kind of more like what these are. So they're very, very energy dense, which is kind of the key to all this. But they're also used to make cholesterols and steroids and other things as the body derives these, as biological systems derive these fatty acids and other types of lipids into varying other forms. But it can have other kinds of biological properties. But because they're called fatty acids, the acid part is obviously a carboxylic acid attached to the end. So this is an example. These typically have carboxylic acid, functionality attached to one end or another, or they're joined together with some kind of, continuous ester-like functionality here. And as they're cleaved apart, these fall apart into carboxylic acids. So that's where the acid part kind of comes into this. But they can have very complex structures. We'll talk a little bit about nucleic acids. These are also acids, carboxylic acids, they're always going to come up that way in organic systems, right? That's your standard kind of organic acid, right? And we'll look at the structures of these, which are made of sugars and an amide base called the nucleoside to form a nucleotide with a phosphate on them. And then they bond together to form these long chains. Similar to proteins, they form these long chains, except that their function in an organism is to store genetic information with the alternating base pairs that are used to store that information. But ribose, these are just carbohydrates, and they're just rings with, alcohol functionalities on them and an ester functionality on them. And again, they color all these to help you see the functional groups, because without that, these don't make a whole lot of sense, right? So we've got all these amine groups, we've got this carbonyl, oxygen, and this helps to explain the properties, right? Part of how these base pairs stick together when the strands of DNA stick together is through hydrogen bonding from the base pairs fitting into each other so that they have enough hydrogen bonding partners to represent each of the pairs. And really what DNA breaks down is to two complementary molecules where one side contains one side of the base pair set and the other side contains the other, and they stick to each other and they twist into each other. So they're two molecules stuck to each other. And they've got other helpers that hold the set together, but that's basically what it breaks down to. In organisms like us, like in eukaryotes, in bacteria, they can have their nucleic acids as like a ring, where the two ends are bonded together and it's just one continuous ring. Part of it is keeping these long, complex structures stable for living things because they store such vital information. And if we think about them as just simply being organic molecules, they're subject to all kinds of oxidative decay and various other side reactions, and that helps us understand how mutation could occur. These are just chemical compounds susceptible to all kinds of side reactions and light and heat and all kinds of varying things. So keeping all that in mind, they got to talk about the nucleoside versus the nucleotide, 5' and 3' end for the positioning to read these. Again, I'm not going to delve into a lot of detail here, but the key that I really want to focus on is this hydrogen bonding that holds the pairs together. Note that G and C get 3 hydrogen bonding sets, and when they're aligned, right, the acceptors and the donors are organized in such a way that they're able to line up physically with each other. So it would be hard for a T to stick to a C because they wouldn't have 3 hydrogen bonding partners for the C, and it would only have 2 for the A. So you can see how this system helps to keep them from getting misaligned. The hydrogen bonding between the 2 strands helps to provide a sort of redundancy for the system, because cells have lots of constantly running housekeeping and repair mechanisms where anytime something might get broken or an amine group gets knocked off or something gets broken in this molecule, they can go back in and repair it as long as they can identify what it should have been, and having the opposite side still be intact can be really useful for that. So this is a way that living systems can help to retain their genetic information against random interaction or side reactions. They say adenine and thiamine form 2 strong hydrogen bonds with each other, but not with guanine and cytosine, as an example. And this is how the helix forms its 2 separate molecules, and they're bonded together with hydrogen bonds at each little interval. And that creates this weaved 2 molecules that store the information, with the information being at the interior. And again, this sugar strands help to provide protection for that chain of information. They say what sequence of bases is complementary. So the original, they form this, even though the sequence is replaced, this goes with G. So they write here as the complementary one. So C goes with G, G goes with C, A goes with T. So you could figure it out from one side. Either one side they call it the sense, and one side it's called the antisense. I'm not going to go into the details about that here. So here they talk about natural versus synthetic products. This is really important in organic. A lot of organic chemistry was driven by the desire of people to make things that previously they could only obtain from some kind of plant or from some kind of mineral that was hard to obtain. And one of the initial driving forces for organic chemistry was to synthesize those natural products, to make them from other things, and to end up with the bioactive thing that someone had wanted, like a synthetic opium or a synthetic, here they talk about a synthetic vitamin C. But the structures for these took a long time to determine because they didn't have good methodologies today like mass spectrometry and other things to be able to figure out these structures. But being able to make them was a driving force of organic chemistry. And so we talk about synthetic. So typically when we talk about a synthetic organic compound, we're talking about one that's made from petroleum byproducts. One of the key uses of petroleum byproducts outside of using them in combustion is to use them to make the medicines and plastics and other kinds of materials that we rely on in our everyday lives. And these are often based on inspiration from natural products. And sometimes we just couldn't grow enough of the plant, or we couldn't make enough of something, or we've modified it in some way to make it more potent, but we started with that as a base in a lot of cases. So sometimes we can distinguish between a natural product and a synthetic one based on the fact that one is from a petroleum source. So it won't have any radioactive carbon in it because all the carbon is carbon-12 at that point. So there are sometimes ways to distinguish it, but chemically they're not any different. So there's not really people talk about how like, oh, well, the natural might be better. Well, not in this sense. It doesn't really matter to biological systems whether the vitamin C was derived from petroleum, but I made it the same structure and it was identical, to one that came from the plant. Now, there might be other compounds in the plant that might be good to have alongside the vitamin C. Maybe there's a synergistic effect that's missing. So I'm not trying to totally dismiss this, but the point of the text here is to say, like, vitamin C is vitamin C. If it has this structure and this formula and these things, then it is that. And it doesn't matter where these individual atoms came from. They could come from the surface of Mars for all we care. They are the same because they are vitamin C. They are ascorbic acid. They fit this structure. They are that same compound. So that's important. And sodium chloride, right? Sodium chloride's the same. There's no chemical distinction between like the rock salts and different kind of salts from the standpoint of the sodium chloride. They might add other things to it to make it different colors, or they might do different things to it, and maybe you don't want those things. But sodium chloride is sodium chloride. Now, you could have different isotopes of the sodium and the chlorine, but on Earth, all those isotopes are not distinguished by biological systems, so those don't even really matter. So the sodium chloride produced by reacting sodium and chlorine in a laboratory is the same as the one found in the ocean. There's no difference between those from a chemical standpoint. All right, well, thanks so much for coming on this journey with me through this chapter. I hope you all, wish you all the best of luck on your tests, and I hope to see you all again in some form or fashion. This is the last lecture for this course, so I wish you all the best of luck on this and the final exam and everything else. Wish you success in the class, and you take care. This is Lucas signing off. Thank you. Thank you for joining me on all this. Take care.